Welcome viewers. Today we are going to discuss how to execute the experiment induction of enzyme activity in E. coli, the lactose operon. Before starting the topic, let's highlight on the important features of bacteria. Bacteria are really very good in adapting to the environment where they are residing. This extraordinary behavior of bacteria is because of the presence of very important regulatory mechanisms in protein biosynthesis. They are very much selective regarding the protein biosynthesis mechanisms. These studies were initially initiated by a group of eminent scientists. The first important contribution was done by Emil Duclos. He was basically working on fungus Aspergillus. What he found that the enzyme invertase was synthesized by this fungus Aspergillus only in presence of its substrate sucrose. Second important contribution in this line was done by Frederick Deinert. He was working on certain yeast cells and found that the enzymes which are needed for the breakdown of galactose were synthesized by the yeast cells only when they were grown in presence of galactose. After that, a series of researches were done. The most important contribution was done by H. Karsten in the year 1930, wherein he for the first time divided the enzymes that are being synthesized by the bacteria into two important categories. Category 1 contained the enzymes which are synthesized by the bacteria throughout its life cycle. The genes that synthesize those proteins were called as constitutive genes or housekeeping genes. Second class of enzymes were called as adaptive enzymes, means they will be synthesized by the organisms only when their expression was needed. Taking these important discoveries and keeping these findings in his mind, two important scientists emerged as Francois Jacob and Jacques Monod. They started working for the first time with E. coli and found that the enzyme called beta-galactosidase, which is needed for the breakdown of lactose, is synthesized by the bacterial cell E. coli only when they were grown in presence of lactose. Later on, they were capable of giving the new concept of operons, which was very much important discovery in terms of understanding the molecular mechanisms or gene regulations going on inside any bacteria. They also gave important clue that when the cells were not grown in presence of lactose or when lactose was not present in the media, then there is no synthesis of beta-galactosidase. So there must be some other regulatory protein that is restricting the synthesis of beta-galactosidase in absence of lactose. And they named it repressor protein. Although they couldn't purify the protein, then came these two important scientists, Walter Gilbert and Benno Muller-Hill. They were successful in purifying the repressor protein. So this is the important contributions that led us to understand the biology of gene regulation in bacteria. So when a microbe or a bacterial cell is residing inside an environment, it has to take certain regulatory decisions. The regulatory decisions depends on two factors. It means it can synthesize two varieties of enzymes. Some enzymes are involved in breakdown of certain substrates. They were called catabolic enzymes. And there were a second class of enzymes that were involved in the synthesis of important metabolites or substrates. Such enzymes were called as anabolic enzymes or biosynthetic enzymes. So the cell will take the decision of the synthesis of catabolic enzymes and biosynthetic enzymes depending on certain factors. If I focus on the catabolic enzyme synthesis, then I will see that the enzymes will be synthesized only when the substrate is present, then the synthesis goes on. On the other hand, if the enzymes are of the biosynthetic pathway, then if the end product of the pathway is present in the growth media, then the synthesis of the operon or the synthesis of these enzymes is switched off. Apart from this, there exists a global regulation in case of catabolic enzymes, wherein we will see that if bacteria finds that there are certain other substrates available in the system which can be metabolized or which can be utilized without the synthesis of so many enzymes, then it will prefer to go for that pathway and it won't synthesize the enzymes needed for the substrate. Operon model states that there are multiple structural genes that are residing under the control of a single promoter. So here we will see that from a single promoter we get a polycistronic mRNA.
Ma'am, what is polycistronic mRNA?
Basically, cistron means one gene. Polycistron means multiple genes. It means once this polycistronic mRNA participates in translation, it will code for more than one protein. So this was the basic principle behind the Operon concept, which is available only in case of prokaryotic system. In eukaryotes, we won't find polycistronic messages. So let's have a look on the negative control of the inducible gene. Here we will see that the repressor is capable of binding onto a regulatory DNA element called operator. Once it binds to the operator, it restricts the binding of RNA polymerase onto this promoter region. Thereby, it switches off the gene expression. On the other hand, as soon as the inducer molecule is coming into the system, it binds to the repressor and changes the conformation of the repressor. As the conformation changes, it loses its binding affinity towards the operator, and as the operator site is vacant now, it can be occupied by RNA polymerase, and RNA polymerase carries on the expression of the gene. Next, look into the negative control of a repressible gene. Means the genes which are getting repressed, means it is the case of a anabolic operon. Herein we will see that repressor molecule alone, which is called as an aporepressor, is incapable of binding to the operator region. So aporepressor and converts it into a functional repressor. So the complex between aporepressor and corepressor then binds to the operator region and restricts the synthesis, as the substrate is already available in the system. So the operon has to be switched off. Next we will see regarding the positive control of an inducible system. In positive control, we will see that there are certain other regulatory elements which are called activators that actually bind to the DNA elements and facilitates the movement of RNA polymerase from the promoter. So what is happening here, that activator binds on the activator binding site, and its binding is promoting the RNA polymerase to move on and carry on the transcription. So the activator can activate the gene expression only when inducer comes in and binds to the activator. In absence of inducer, the activator remains alone. It cannot activate the process of transcription. So next, if we talk about a positive control of a repressible system, herein we have seen that the activator protein alone can bind onto the system and promotes RNA polymerase to carry on the transcription. On the other hand, as soon as the end product comes into the system, it binds to the activator and removes it from the activator binding site, thereby restricts the transcription pathway. So, if I focus precisely on the lactose operon, which is our topic of discussion today, we will see that lac operon comprises of three structural genes, namely LAC-Z, LAC-Y and LAC-A, wherein LAC-Z codes for beta-galactosidase enzyme, which is capable of breakdown of lactose into galactose and glucose moieties, as these two components are linked together by a linkage called beta-galactosidic linkage, which is cleaved by beta-galactosidase enzyme. Second enzyme is lactose permease. It actually allows the entry of the lactose molecules inside the bacterial cell, and the function of transacetylase is yet to be discovered. Apart from this, in the upstream region of the structural genes, we have got the regulatory elements, namely operator site, promoter element and cap binding site. In its further upstream, we have got the LAC-I gene that codes for LAC repressor. The Lac repressor is being synthesized by a promoter called Lac-I, which is a constitutive promoter. As I said, a constitutive promoter keeps on synthesis of Lac-I throughout the life cycle of the bacteria. If the bacteria is growing in an environment where there is no lactose, under such circumstances, bacteria really don't need to synthesize the enzymes which are needed for the breakdown of lactose. It means the operon has to remain switched off under such circumstances. So who is going to switch off the operon? See, the constitutive synthesis of Lac repressor leads to the formation of dimer, and this dimer binds to the Lac operator, which is the regulatory DNA element. Once it binds to the Lac operator, it restricts the binding of RNA polymerase onto the Lac promoter. And if it cannot bind to the Lac promoter, then there is no synthesis of the structural genes. Next, what happens into the cell when lactose comes into the environment via the lactose permease? Herein, we will see that lactose gets isomerized into allolactose, which is acting as an inducer. It binds to the repressor and changes its three-dimensional conformation, and as a result of which, it no longer binds to the Lac operator. As Lac operator site is vacant, now RNA polymerase will come, bind to the promoter, carry on the synthesis of the structural genes, leading to the synthesis of beta-galactosidase, which is going to break down lactose. As it is needed, as soon as lactose comes, it is being broken down by beta-galactosidase. So this is all about the negative regulation of lactose operon.
Ma'am, what happens when E. coli is allowed to grow in a medium which is both glucose and lactose?
Yes, this is the point where the bacteria really has to take certain decisions. To understand this thing properly, let's have a look on the positive control of Lac operon. Then all your queries will be fulfilled. Let's take different conditions. If the bacteria is grown in presence of lactose, but there is no glucose. If lactose is there, it means repressor has been inactivated by the inducer molecule allolactose. On the other hand, if glucose level is low, then it is inversely proportional to the concentration of cyclic AMP. It means cyclic AMP level is high. Then cyclic AMP CRP complex or CAP complex binds to an activator site called CAP binding site. And it is acting as a positive regulator, and what it does, it positively regulates the movement of RNA polymerase which is bound onto the promoter region. Thereby, it increases the rate of synthesis of the structural genes of Lac operon, leading to breakdown of lactose. Second condition, as you were asking, is what happens if both lactose and glucose are present. If lactose is present, then repressor molecule is definitely being removed from the operator side. On the other hand, as I said, that if glucose level is high, then the cyclic AMP level is low, and there is no complex formation between cyclic AMP and CAP. And as a result of which, the CAP site, which is the positive activator binding site, remains vacant. So, the promoter region of the Lac is not strong enough to carry on the transcription of the structural genes alone just by removal of the repressor molecule from the operator site. So here we will see that transcription is inhibited due to the lack of cyclic AMP CRP complex, right? Next, think of the situation when there is neither glucose nor lactose. Then we will see that if glucose is not present, then cyclic AMP CRP, though it is binding onto the CAP site, but the operator site is being occupied by the repressor molecule. So no question of transcription. Operons remain switched off. Next is if glucose and lactose both are present. So what will happen if lactose is not present? Then repressor will bind to the operator, and glucose is present. It means cyclic AMP CRP is not binding onto the CAP site. So definitely the operon is remaining switched off because of this problem. Now we are coming into the experimental part where we are going to measure the production of the enzyme beta-galactosidase by E. coli using ONPG assay.
Ma'am, what is ONPG and what is the principle behind this assay?
Actually, ONPG stands for ortho nitrophenol beta galactoside. Normally, lactose comprises of a galactose moiety and a glucose moiety, which is broken down by beta-galactosidase enzyme to produce glucose and galactose. In ONPG, we also have a galactose moiety, which is being attached to an orthonitrophenol group via beta-galactosidic linkage. And it is again being broken down by beta-galactosidase. If E. coli produces beta-galactosidase, then this linkage will be broken and we will get back again a galactose molecule and orthonitrophenol. This orthonitrophenol imparts yellow color, which can be measured spectrophotometrically. So the amount of orthonitrophenol is directly proportional to the amount of breakdown of ONPG, or in other words, the amount of beta-galactosidase that is getting synthesized by induction of Lac operon. The materials which are needed to execute the experiment is a strain of E. coli B, which has got Lac operon active. Next, we have got orthonitrophenyl beta D galactoside. It has to be dissolved in phosphate buffer with pH 7, as this pH is necessary for the activity of the enzyme beta-galactosidase. Next, we need methyl benzene or toluene for the lysis of the cells, or to make the cells permeable so that ONPG gets entry inside the bacterial cells. Then we need sodium carbonate to stop the reaction. And the media in which we will be growing E. coli is a minimal media, which is called as Cold Spring Harbor A media. Then we need test tube, cap, racks, inoculation loop, pass-through pipettes, and water bath, which is to be maintained at 37 degrees centigrade. We need cuvettes for spectrophotometer and a spectrophotometer. Now let's have a look on the qualitative method of estimating the expression of beta-galactosidase. Here, first we will take 2 ml of ONPG in 2 different tubes. After that, we will be adding a loopful of bacteria is taken aseptically from the plate. It is added onto the solution containing ONPG, and it has to be twiddled properly so that the bacteria gets dispersed into the ONPG. After that, we will be adding a drop of methyl benzene to each of the tubes, right? One tube is called as the control, where we haven't inoculated any bacteria, and the second one is the test one, where bacteria has been inoculated. And this entire thing is incubated for 5 to 20 minutes in a 37-degree water bath incubator until a yellow color develops. This yellow color is indication of generation of orthonitrophenol. It means orthonitrophenyl beta-galactoside has been broken down by the enzyme beta-galactosidase, right? So once qualitatively we have estimated that the bacteria is synthesizing beta-galactosidase, we really need to quantitate our result. To execute a quantitative estimation, first the cells should be grown in Cold Spring Harbor media containing either 0.1% glucose, and the second one where we have inoculated it with Cold Spring Harbor media plus 1% of lactose. So the previous one, there will be no expression of beta-galactosidase, as this media contains only glucose, and the second one contains lactose. After overnight grown cultures are taken and they are centrifuged. After centrifugation, we are going to discard the supernatant and will take the pellet. After that, the pellet is being suspended in Z buffer, and it is vortexed properly to make a homogeneous mix of the cells. After mixing and getting a suspension culture of a bacteria, we will be taking the OD of the cells at absorbance of 660 nanometers, so that 5 ml of the culture gives an OD of 0.2. So if needed, proper dilutions are to be made. After this, the next step will be addition of toluene or methyl benzene. This is done after addition of 3 drops of methyl benzene. We transfer the tubes onto the water bath, and before transferring it onto the water bath, 2 ml of ONPG solution is added on each sample. Then It is incubated at 37 degrees centigrade incubator, water bath incubator for 20 minutes. We will see that the tube that was containing cells grown in presence of lactose gave development of yellow color. On the other hand, the tube that was grown in presence of glucose only didn't give any yellow color development. So to stop the reaction, we really need to add 1 molar of sodium carbonate. After that, we will be taking the OD measurement at 420 nanometers. 420 nanometer is to record the yellow color which is developed due to generation of orthonitrophenol. And finally, after getting the result, we really need to calculate the units of beta-galactosidase activity. Here the formula that we are going to use is 1000 is to be multiplied by the OD value that we are getting at 420 nanometers, is to be subtracted with 1.75 times the OD that we are getting at 550 nanometers, and here V stands for the volume of the sample and T stands for the time of incubation, and OD at 600 nanometers gives me an idea that how many number of cells were present as we started the experiment.
Ma'am, why are we subtracting that factor from the OD we have calculated at 420?
Yeah, it's really a very nice question, as the cells are already present when we are recording the OD at 420 nanometers. So the OD that we are getting at 420 nanometers is not only because of the orthonitrophenol, but certain amount of light scattering is also occurring because of the cell debris. So we really need to subtract that. That's why we have taken the OD at 550 nanometers, wherein we can measure the light scattering due to the cells. And if I multiply it by a factor of 1.75, then it becomes equivalent to the OD that we are getting at 420 nanometers for light scattering. So this is to make our result more precise, so that whatever OD we are recording is just because of orthonitrophenol. So while you are performing this experiment, certain precautions are to be taken. As you are performing microbiological experiment, all microbiological safety measures, aseptic techniques are to be opted. Second important thing is, as methyl benzene is flammable and produces harmful vapors, so you have to be very careful. If the amount of methyl benzene that you are using is too much high, then it's better to use fume chambers. And skin and eye contacts should be avoided. If necessary, we can use specs to protect our eyes. Today's lecture, I think you have understood the basic principle of lactose operon and the various gene regulations that actually make bacteria so much suitable to accommodate in the environment. And we have finally highlighted on the tidbits of executing the ONPG assay, that first of all, whenever you execute an experiment, you should do a qualitative assay. There, if you get a positive response, then you will be going for a quantitative assay to really compare the level of expression of beta-galactosidase between different strains of bacteria. So that's all for today.